Good afternoon, everyone. My name is Puhl, and I'm a senior scientist at Collaborations Pharmaceuticals. Thank you, Amber, for inviting me to be here. Such a great opportunity. I'm thrilled to be here. Actually, seeing the faces of people that I have read papers before. So this is so exciting, like to make all these connections. And I hope that this conference will bring novel opportunities and novel treatments or discovery of treatments for SLC6A1. We actually never heard about this disease before until the day that Amber contacted my CEO, Sean Eakins. And then we started studying about this disease and trying to help to find ways that we can approach this problem and hopefully find a treatment for this disease. So we are a small pharmaceutical company. So everyone that you're seeing in the picture actually is the whole company. You can see my CEO, Sean Eakins, in the left. And this company was founded in 2015, and currently we have 9 employees. Up to date, we have 6 FDA orphan drug designations for different diseases, and the company doesn't have any VC or angel capital. It's totally funded by grants. To the moment, we raised $7 million through 16 grants from NIH, 1 DOD, and 1 DITRA funded projects. We focus highly on rare and neglected diseases. In this picture, you can see like some of our diseases that we are in our portfolio. We are working on Ebola. Up to the moment, we developed a drug that can prevent Ebola infection in guinea pigs, and our next stage is to test that in monkeys. We also have compounds for Chagas, tuberculosis, and now other diseases. On the rare disease side, we focus mainly on Batten disease, where we are developing an enzyme replacement therapy for CLN1, and we also have our screening small molecule chaperones for that disease. As you can see here, to the other side, most of our projects involve machine learning techniques, and I'll talk a little bit about this. So the main driving force of our company is machine learning. In the way that this works, we get data from data that are publicly available through Campbell, PubChem, BindingDB, or data that we produce in-house, or that comes from our collaborators. We get this data, curate the data, build models, generate predictions, and perform experiments. What you're seeing on the other side is like statistics from a model. So after we generate the model from this data that is available, we can see if it's a good model and if we can predict novel compounds. Usually, we screen virtually FDA-approved drugs or other libraries of interest. And for neurological diseases, we can apply like a filter to predict if these molecules can cross the blood-brain barrier, and also to predict cytotoxicity. So the way that we operate, we build machine learning models using our software, Assay Central. We cherry-pick some compounds to test. We test them in vitro using several biophysical and biochemical techniques. And if we validate those hits, we move forward to testing them in patient cell lines when available, or we can also outsource for collaborators. Now, moving on to the main topic of today, I'll be combining all the information that has been already presented, like a summary and try to explain the possible approaches that we can take for this disease. So what we know about SLC6A1? We know that the GAT1 knockout mice exhibit spontaneous spike wave discharges and absence seizures. And also, it has been reported that up to 4% of patients with myoclonic atonic epilepsy present SLC6A1 mutations, de novo inactivating mutations, suggesting that these variants might be specific for this myoclonic atonic epilepsy, and this can cause cognitive impairment after seizure onset. But what was pointed today that actually the phenotype is kind of broad. We have absence seizures, we have MIA, and other types of absence seizures. It has been pointed today that the GAT dysfunction might impact the GABA A and GABA B signally in several ways. One of them is that GAT1 dysfunction is expected to reduce the clearance, leading to increased GABA levels both at the synaptic and extrasynaptic. And this can lead to overstimulation of extrasynaptic GABA A and GABA B receptors. So the hypothesis about mechanism that has already been pointed for several speakers today. If we have an increased GABA A, GABA receptor mediating tonic inhibition, we can have a neuronal hyperpolarization and burst pattern firing in thalamocortical neurons, which can promote the generation of the spike discharges. So that was one hypothesis. The other is that we're seeing here that we have the postsynaptic GABA B receptors. And when we have activation of these receptors, we can increase the potassium efflux, but we can also activate low voltage calcium channels, and that might lead to the recurrent excitation within the thalamocortical system through successive sodium spikes. In previous work by Sned in '92, and after he also published other papers in a similar way, showed that GABA B receptor activation causes absence seizures in mice and rats, and that the pretreatment with GABA B antagonists can decrease the duration of these chemically induced absence seizures. So later on, he also published a paper showing a model of atypic absence seizures where the treatment with GABA B antagonists decrease the cognitive impairment and seizures in mice and rats. And also, it was showing today that if we have a reduce of the activity of the GAT1 transporter, this might also impair the phasic signaling of GABA A, because we don't have the uptake of GABA, so maybe we can have less GABA inside of the cells and less release, or these receptors might become desynthesized. So taken together, this was proposed by Mattison and collaborators. If we have an impairment of the GAT1 receptor that is present in the neurons, but also in astrocytes and other cells, if we have an impairment of this transporter, the reduced GAT1 function might lead to epilepsy through overactivation of extrasynaptic GABA-A and GABA-B receptors, and reduction of GABA-A synaptic signaling. So probably this GABA is activating GABA-B extrasynaptically, and this might be causing those spike or discharges by the influx of calcium. So here, my task today is talk about potential drug targets, but actually those are more ideas since we don't have a full knowledge of this disease and it's such a complex mechanism. But based on the hypotheses that were presented in this paper, we can come up with the idea that antagonists of GABA-B receptors might ameliorate the symptoms of this disease. So as it was showing before, if we have an excess of GABA, we can activate GABA-B extrasynaptically. This can activate these proteins, GERK, and promote a flux of potassium, and that will also increase the hyperpolarization. And we can also have an activation of these low voltage calcium channels that they are not selective, and that can cause some spikes. But also, if we look here in this pathway, we are increasing the formation of AMP cyclic, and I think it's not clear what type of other receptors or other proteins can be activated by an excess of AMP cyclic. And there's a connection between the hyperpolarization cyclic nucleotide channels that can become activated by an increase of AMP cyclic. So in that case, those channels, they are also calcium channels, so that might also promote some discharge. But those receptors channels are not present in all areas of the brain, but I think it's interesting to try to look to those other mechanisms behind the disease. And I brought up this paper that is associated with Rett syndrome. Actually, the mutation is in another gene, but actually the mutation decreases the GAT1 transporter. In the knockout mice of GAT1 and MCP2, they display an overlapping phenotype. In this paper, when they inhibit GABA-B receptors, actually they show that GAT1 protein levels were decreased. So we might find some similarities between the GAT1 and the Rett syndrome. And they show when they use GABA-B receptor antagonists, they actually decrease the cortical epileptiform discharges. So I think this, again, gives another indication that now that we have all these available mice models and cell models and astrocytes and everything that was presented here, that we can try to use some GABA-B antagonists to verify the impact that they will have on the disease. And as I mentioned before, GABA-B antagonists also abolish atypical absence seizures. I'll skip this one. I would like to talk about the possibility to enhance the GAT1 activity. This might not be possible for all the mutations in SLC61, but it has been shown that some mutations display up to 33% of activity. And Caddy just published a paper where this patient that has 33% of activity of GAT1 develop seizures much later. So maybe if we can rescue some of the activity of the GAT1, we can also have an impact in this disease. And it has been shown, like in this paper, there's some receptors that are coupled to G proteins that they work in both ways. When we have low adenosine in the cells, they inhibit GABA uptake. And when we have high adenosine in the cells, they increase GAT1 activity. So it's possible to modulate the activity of the receptor. I only have 1 minute left, and I have so much to say. So this is one possibility. So I'll just move forward to the next one. It was shown that in phasic synaptic, we don't have much. We might have a. My idea is that it might be possible to also use inhibitors of vesicular GABA transporter, so not much GABA will be released. So it will be less GABA in the synaptic cleft, and this can also modulate. These are hard. There's no many compounds out there, but it might be an option for a screening. There are several questions that we need to address because we don't know the levels of GABA in the synaptic cleft, how much we need to decrease or modulate, and what is happening in this disease, what receptors are upregulated or downregulated that can play a role. We are currently collaborating with PhD, and we have 2 compounds that we would like to test in cultured neurons and astrocytes, and further in animal models. We are aiming to continue to study and propose targets, use machine learning models to find compounds, and test and see if we can help to develop a treatment for this disease. With that, I'd like to thank you all for your attention, and I'm open to questions.
Yeah, that was really terrific. I just wanted to reinforce something I said in response to Caddy's question that might get people thinking a bit. And I think your idea about using GABA B antagonists is really a great one. This has a long history. And the pharmaceutical industry, as I mentioned, had worked on this issue for a long period of time. The company that was particularly involved is a company called Ciba Geigy, and they no longer exist by themselves. They're part of a big Swiss company called Novartis. And they had been working on the idea of GABA B antagonists for absence epilepsy. But because of what I think was just a business decision, they never pursued that. And the reason they didn't pursue it from a business point of view is that we already have plenty of good drugs for absence epilepsy. And they didn't think there would really be a need for it. And I tend to agree with them on that. But we're in a new era where we've identified these new types of epilepsies, right? And we can identify them genetically. And these targeted drugs that we put to the side previously, we should think about bringing them back, testing them in the animal models, and then if there's some indication of activity, we could encourage people to invest in those drugs again for these targeted rare conditions. So I think that strategy is really a very promising one.
Thank you.
I just want to follow Dr. Rogaski, make a short comments. So I look at, actually after you guys visit my lab, I look at a GABA B blocker. Actually, few of them are brain permeable. So if they are brain permeable and we have a mouse model available, we can directly test. Yeah.
Let's do it.
Yeah, I think with all these models, we can also evaluate what's happening, because through the literature, we can only hypothesize, but I think now that the models are available, we can verify the expression of all the proteins involved in the GABAergic pathway, like to see if the transaminases are increased, if the carboxylase is decreased, or if we have more receptors or less in the synaptic cleft. I think that will address a lot the direction that we might take to approach this disease. Okay, thank you very much.